Good morning, it's Dr. Will, CBMD, PhD. It is Thursday, November 10, 2011, 2:51 a.m. Eastern Time. Quick note. Easily reprogrammable cells could be key in creation of new life forms. Science Daily, November 7, 2011. Scientists at the University of North Hagen are leading an ambitious research project to develop a viable biological cell equivalent of a computer operating system. The success of the project to create a reprogrammable cell could revolutionize synthetic biology and would pave the way for scientists to create completely new and useful forms of life using relatively hassle-free approach. And good morning, it's Dr. Will, CBMD, PhD. It is Thursday, November 10, 2011, 2:52 a.m. Eastern Time. Quick note. We are talking about a highly ambitious goal leading to a fundamental breakthrough that will ultimately allow us to rapidly prototype, implement, and deploy living entities that are completely new and do not appear in nature, adapting them so they perform new useful functions. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 2:53 a.m. Eastern Time. Quick note. The changing technology could substantially accelerate synthetic biology research and development, which has been linked to myriad applications, from the creation of new sources of food and environmental solutions to a host of new medical breakthroughs, such as drugs tailored to individual patients and the growth of new organs for transplant patients. And good morning, it's Dr. Will, CBMD, PhD. This Thursday, November 10, 2011, 2:56 a.m. Eastern Time. Quick note. The project towards biological cell operating systems, audacious, is attempting to go beyond system biology to science behind understanding how living organisms work to give scientists the power to create biological systems. The scientists will start the work by attempting to make E. coli bacteria much more easy to program. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 2:58 a.m. Eastern Time. Quick note. Currently, each time we need a cell that will perform a certain new function, we have to relocate it from scratch, which is a long and laborious process. Most people think all we have to do to modify behavior is to modify a cell's DNA, but it is not as simple as that. We usually find we get the wrong behavior and then we are back to square one. If we succeed with this audacious project in 5 years' time, we will be programming bacterial cells in the computer and compiling and storing its program into these new cells so they can readily execute them. And good morning, it's Dr. Will, CBMD, PhD. It is Thursday, November 10, 2011, 3:01 a.m. Eastern Time. Quick note. Like for a computer, we are trying to create a basic operating system for a biological cell. Among the most fundamental challenges facing the scientists will be developing new computer models that will more accurately predict the behavior of cells in the laboratory. Scientists can already program individual cells to complete certain tasks, but scaling up to create a larger organism is trickier. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 3:01 a.m. Eastern Time. Quick note. The creation of more sophisticated computer modeling programs and a cell that could be reprogrammed to fulfill any function without having to go back to the drawing board each time could largely remove the trial and error approach currently taken and allow synthetic biological research to take a significant leap forward. And good morning, it's Dr. Will, CBMD, PhD. This Thursday, November 10, 2011, 3:02 a.m. Eastern Time. Quick note. The creation of more sophisticated computer modeling programs in the cell that could be reprogrammed to fulfill any function without having to go back to the drawing board each time could largely remove the trial and error approach currently taken and allow synthetic biology research to take a significant leap forward. The technology could be used in a whole range of applications where being able to modify the behavior of organisms could be advantageous. In the long run, this includes the creation of new microorganisms that could help to clean the environment, for example by capturing carbon from the burning of fossil fuel or removing contaminants, arsenic from the water sources. Alternatively, the efficiency of medicine could be improved by tailoring it to specific patients to maximize the effect of the drugs and to reduce any harmful side effects. And good morning, it's Dr. Will, CBMD, PhD. It is Thursday, November 10, 2011, 3:05 a.m. Eastern Time. Quick note. NASA developed super black material that absorbs light across multiple wavelength bands. Science Daily, November 8, 2011. NASA engineers have produced a material that absorbs, on average, more than 99% of the ultraviolet visible infrared and far infrared light and hits it. A development that promises to open new frontiers in space technology. And good morning, it's Dr. Will, CBMD, PhD. It's Tuesday, November 10, 2011, 3:06 a.m. Eastern Time. Quick note. The team of engineers at NASA Goddard Space Flight Center, Greenbelt, MD, reported their finding recently at the SPI Optics and Photonics Conference, the largest interdisciplinary technical meeting in this discipline. The team has since reconfirmed the material's absorption capabilities and additional testing, who is leading to the effort involving 10 Goddard Technologies. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 3:08 a.m. Eastern Time. Quick note. The reflectance test showed that our team had extended by 50 times the range of the material's absorption capabilities. Though other researchers are reporting near perfect absorption levels, mainly in the ultraviolet and visible, our material is darn near perfect across multiple wavelength bands, from the ultraviolet to far infrared. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 3:08 a.m. Eastern Time. Quick note. No one else has achieved this milestone yet. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 3:13 a.m. Eastern Time. Quick note. The nanotech-based coating is a thin layer of multi-walled carbon nanotubes, tiny hollow tubes made of pure carbon about 10,000 times thinner than a strand of human hair. They are positioned vertically on various substrate materials, much like a shag rug. The team has grown the nanotubes on silicone nitride, titanium, and stainless steel materials commonly used in space-based scientific instruments to grow carbon nanotubes. Goddard technologist Stefanie Getty applies a catalyst layer of iron to an underlayer on silicone, titanium, and other materials. She then heats the material in an oven to about 1,382 degrees Fahrenheit. While heating, the material is bathed in carbon-containing feedstock gas. And good morning, it's Dr. Will, CBMD, PhD. Thursday, November 10, 2011, 3:17 a.m. Eastern Time. Quick note. The tests indicate that the nanotube material is especially useful for a variety of spaceflight applications where observing in multiple wavelength bands is important to scientific discovery. One such application is stray light suppression. The tiny gaps between the tubes collect and trap background light to prevent it from reflecting off surface and interfering with the light that scientists actually want to measure. Because only a small fraction of light reflects off the coating, the human eye and sensitive detectors see the material as black. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 3:22 a.m. Eastern Time. Quick note. In particular, the team found that the material absorbs 99.5% of the light in the ultraviolet and visible, dipping to 98% in the longer or far infrared bands. The advantage over other materials is that our material is from 10 to 100 times more absorbent, depending on the specific wavelength band. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 3:24 a.m. Eastern Time. Quick note. If used in detectors and other instrument components, the technology would allow scientists to gather hard to obtain measurements of objects so distant in the universe that astronomers no longer can see them in visible light, or those in high contrast areas, including planets in orbit around other stars. Earth scientists studying the oceans and atmosphere also would benefit. More than 90% of the light Earth monitoring instruments gather comes from the atmosphere, overwhelming the faint signal they are trying to retrieve. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 3:28 a.m. Eastern Time. Quick note. Currently, instrument developers apply the paint baffles and other components to help prevent stray light from ricocheting off surfaces. However, black paints absorb only 90% of the light that strikes it. The effect of multiple bounces makes the coating's overall advantage even larger, potentially resulting in hundreds of times less stray light. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 3:31 a.m. Eastern Time. Quick note. In addition, black paints do not remain black when exposed to cryogenic temperatures. They take on a shiny, slightly silver quality. Who is evaluating the carbon nanotube material for use as a collaborator on far infrared sensing instruments that must operate in super cold conditions to gather faint far infrared signals emanating from objects in the very distant universe. If these instruments are not cold, thermal heating generated by the instrument and observatory will swamp the faint infrared they are designed to collect. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 3:35 a.m. Eastern Time. Quick note. Black materials also serve another important function on spacecraft instruments, particularly infrared sensing instruments. The blacker the material, the more heat it radiates away. In other words, super black materials like a carbon nanotube coating can be used on devices that remove heat from instruments and radiate it away to deep space. This cools the instruments to lower temperatures where they are more sensitive to faint signals. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 3:37 a.m. Eastern Time. Quick note. To prevent the black paints from losing their absorption and radiative properties, a long wavelength instrument developers currently use epoxy loaded with conductive metals to create a black coating. However, the mixture adds weight, always a concern for instrument developers. With the carbon nanotube coating, however, the material is less dense and remains black without additives, and therefore is effective at absorbing light and removing heat. This is a very promising material. It is robust, lightweight, and extremely black. It is better than black paint by a long shot. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 4:55 a.m. Eastern Time. Quick note. Half-billion-year-old predator-tracked multi-leg creature ruled the seas. Science Daily, November 8, 2011. Researchers from the University of Saskatchewan Royal Ontario Museum, ROM, have followed fossilized footprints to a multi-leg predator that ruled the seas the Cambrian period about half a billion years ago. Short of finding an animal at the end of the trackway, it's really very rare to be able to identify the producer so confidently. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 5:01 a.m. Eastern Time. Quick note. The research team worked with samples gathered from the Burgess Shale, famed for its exquisitely detailed fossil from the Cambrian explosion, a time when life underwent a dramatic change with the appearance of all the modern groups of organisms and some bizarre creatures, located near the village of the Field in Canada's Yahoo National Park in British Columbia. The Burgess Shale is an international treasure, providing an unparalleled window into the distant past. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 5:04 a.m. Eastern Time. Quick note. Ancient lunar dynamo may explain magnetized moon rocks. Science Daily, November 9, 2011. The presence of magnetized rocks on the surface of the moon, which has no global magnetic field, has been a mystery since the days of the Apollo program. Now a team of scientists has proposed a novel mechanism that could have generated a magnetic field on the moon early in its history. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 5:09 a.m. Eastern Time. Quick note. The geodynamo that generates Earth's magnetic field is powered by heat from the inner core, which drives complex fluid motions in the molten iron of the outer core. But the moon is too small to support that. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 5:10 a.m. Eastern Time. Quick note. Technology described how an ancient lunar dynamo could have arisen from stirring of the moon's liquid core driven by the motion of the solid mantle above it. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 5:11 a.m. Eastern Time. Quick note. This is a very different way of powering a dynamo that involves physical stirring, like stirring a bowl with a giant spoon. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 5:12 a.m. Eastern Time. Quick note. Calculated the effects of differential motion between the moon's core and mantle. Early in its history, the moon orbited Earth at a much closer distance than it does today, and it continues to gradually recede from Earth. At close distances, tidal interactions between Earth and the moon cause the moon's mantle to rotate slightly differently than the core. The differential motion of the mantle relative to the core stirred the liquid core, creating fluid motions that, in theory, could give rise to a magnetic dynamo. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 5:15 a.m. Eastern Time. Quick note. The moon wobbles a bit as it spins. That's called precession. But the core is liquid, and it does do exactly the same precession. So the mantle is moving back and forth across the core, and that stirs up the core. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 5:16 a.m. Eastern Time. Quick note. The researchers found that the lunar dynamo could have operated in this way for at least a billion years. Eventually, however, it would have stopped working as the moon got farther away from Earth. The further out the moon moves, the slower the stirring. And at a certain point, the lunar dynamo shuts off. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 5:19 a.m. Eastern Time. Quick note. Rocks can become magnetized from the shock of an impact, a mechanism some scientists have proposed to explain the magnetization of lunar samples. But recent paleomagnetic analysis of moon rocks, as well as orbiting measurements of the magnetization of a lunar crust, suggest that there was a strong, long-lived magnetic field on the moon early in its history. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 5:24 a.m. Eastern Time. Quick note. One of the nice things about our model is that it explains how a lunar dynamo could have lasted for a billion years, Nemo said. It also makes predictions about how the strength of the field should have changed over the years, and that's potentially testable with enough paleomagnetic observations. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 5:31 a.m. Eastern Time. Quick note. More detailed analysis is needed, however, to show that stirring of the core by the mantle would create the right kind of fluid motions to generate a magnetic field. Only certain types of fluid motions give rise to magnetic dynamos. We calculated the power that's available to drive the dynamo and the magnetic field strengths that could be created. But we really need the dynamo experts to take this model to the next level of detail and see if it works. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 5:35 a.m. Eastern Time. Quick note. A working model of a lunar dynamo combined with more detailed paleomagnetic analysis, the moon rocks could give scientists a powerful tool for investigating the history of the moon. In addition, the study presents a novel mechanism for generating a magnetic field not only on the moon but also on the small bodies, including large asteroids. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 5:50 a.m. Eastern Time. Quick note. Do plants perform best with family or strangers? Researchers consider social interactions. Science Daily, November 9, 2011. In the fight for survival, plants are capable of complex social behaviors and may exhibit altruism towards family members but aggressively compete with strangers. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 5:58 a.m. Eastern Time. Quick note. A growing body of work suggests plants recognize and respond to the presence and identity of their neighbors. But can plants cooperate with their relatives? While some studies have shown that siblings perform best, suggesting altruism towards relatives. Other studies have shown that when less related plant growth together, the group can actually outperform siblings. This implies the group benefits from its diversity by dividing precious resources effectively and competing less. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 6:01 a.m. Eastern Time. Quick note. A team from McMaster University suggests plants can benefit from both altruism and biodiversity, but when these processes occur at the same time, it is difficult to predict the outcome. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 6:01 a.m. Eastern Time. Quick note. The greatest challenge for understanding plant social interactions is we can't interpret plant behaviors as easily as we do those of animals. Though we have shown plants change traits in the presence of relatives, we need to determine if this is cooperation. Linking the plant behaviors with their benefits is challenging when multiple processes co-occur. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 6:02 a.m. Eastern Time. Quick note. The problem is that plant social interactions are treated as a black box, with researchers only looking at the output or the fitness of the plant. In sibling competition, but they need to investigate the mechanisms inside the box by describing how traits of individuals affect fitness, to understand how the output is reached and which mechanisms are occurring to get there. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 6:04 a.m. Eastern Time. Quick note. Simply put, social environment matters to plants. If we first acknowledge that the kin cooperation and resource partitioning are co-occurring, we can begin to address some very important questions. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 6:04 a.m. Eastern Time. Quick note. Among these questions is whether there is a link between kin recognition and plant performance, whether plant kin recognition can improve crop yield, and how kin recognition shapes communities and ecosystems. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 6:34 a.m. Eastern Time. Quick note. Methane may be answered a 56-year-old question. Ocean could have contained enough methane to cause drastic climate change. Science Daily, November 9, 2011. The release of massive amounts of carbon from methane hydrate, frozen under the seafloor 56 million years ago, has linked to the greatest change in global climate since a dinosaur-killing asteroid presumably hit Earth 9 million years earlier. New calculations by researchers at Rice University show that this long and controversial scenario is quite possible. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 6:35 a.m. Eastern Time. Quick note. Nobody knows for sure what started the incident, but there is no doubt Earth's temperature rose by as much as 6 degrees Celsius. That's affected the planet for up to 150,000 years until excess carbon in the oceans and atmosphere was reabsorbed into sediment. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 6:36 a.m. Eastern Time. Quick note. Earth's ecosystem changed and many species went extinct during the Paleo-Eocene thermal maximum, PETM, 56 million years ago, when at least 2,500 gigatons of carbon eventually in the form of carbon dioxide were released into the ocean and atmosphere. The era is described in great detail in a recent National Geographical feature. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 6:37 a.m. Eastern Time. Quick note. In a report like Rice Scientist, Nature, Geoscience suggests that at the time, even though methane-containing gas hydrates, the ice that burns occupied only a small zone of sediment under the seabed before the PETM, there could have been as much stored then as there is now. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 6:39 a.m. Eastern Time. Quick note. This is a concern to those who believe the continued burning of fossil fuels by humans could someday trigger another feedback loop that disturbs the stability of methane hydrate under the ocean and in permafrost. This change could warm the atmosphere and prompt the release of large amounts of methane, a more powerful greenhouse gas than carbon dioxide. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 6:41 a.m. Eastern Time. Now, some who study the PETM blame the worldwide burning of peat, volcanic activity, or a massive asteroid strike as the source of the carbon. But there is no creator or any suit or evidence of burning of peat. And Good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 6:42 a.m. Eastern Time. Quick note. In the ocean, organisms die, sink into the sediment, and decompose into methane. Under high pressure and low temperatures, methane molecules are trapped by water, which freezes into slushy substance known as gas hydrate that stabilize in a narrow band under the seafloor. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 6:43 a.m. Eastern Time. Quick note. Warmer oceans before the PETM would have made the stability zone for gas hydrate thinner than today, and some scientists have argued this would allow for much less hydrate than exists under the seafloor now. If the volume the size of the box was less than today, how would it have released so much carbon? And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 6:45 a.m. Eastern Time. Quick note. The critics said, "No, this can't be. It's warmer. There couldn't have been more methane hydrate." But we applied the numerical model and found that if the oceans were warmer, they would contain less dissolved oxygen, and the kinetics for methane formation would have been faster. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 6:46 a.m. Eastern Time. Quick note. With less oxygen to consume organic matter, on the way down, more sank to the ocean floor. With seafloor temperatures higher than they are today, microbes that turn organic matter into methane work faster. Heat speeds things up. It is true for almost all microbial reactions. That's why we have refrigerators. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 6:46 a.m. Eastern Time. Quick note. The result is that a stability zone smaller than what exists now may have held a similar amount of methane hydrate. We are increasing the feedback stock, processing it faster, and packing it in over what could have been millions of years. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 6:49 a.m. Eastern Time. Quick note. While the event that began the carbon discharge cycle remains a mystery, the implications are clear. I've always thought of the hydrate layer as being like a capacitor in a circuit. It charges slowly and can release fast, and warming is the trigger. It's possible that's happening right now. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 6:51 a.m. Eastern Time. Quick note. That makes it important to understand what occurred in the PETM. The amount of carbon released then is on the magnitude of what happens will add to the cycle by the end of, say, 2500. Compared to the geological timescale, that's almost instant. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 6:52 a.m. Eastern Time. Quick note. We run the risk of reproducing that big carbon discharge event, but faster by burning fossil fuel. And it may be severe if hydrogen dissociation is triggering again. That methane hydrate also offers the potential to become a valuable source of clean energy, as burning methane emits much less carbon dioxide than other fossil fuels. The calculation should encourage geologists who discounted hydrates' impact during the PETM to keep an open mind. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 7:21 a.m. Eastern Time. Quick note. Greenhouse gas index continues to climb. Science Daily, November 9, 2011. NOAA's updated annual greenhouse gas index, AGGI, which measures the direct climate influence of many greenhouse gases such as carbon dioxide and methane, shows a continued steady upward trend that began with the Industrial Revolution of the 1880s. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 7:24 a.m. Eastern Time. Quick note. Started in 2004, the AGGI reached 1.29 in 2010. That means the combined heating effect of long-lived greenhouse gases added to the atmosphere by human activities has increased by 29% since 1990. The index year used as a baseline for comparison. This is slightly higher than 2009 AGGI, which was 1.27 when the combined heating effect of those additional greenhouse gases was 27% higher than in 1990. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 7:28 a.m. Eastern Time. Quick note. The increasing amount of long-lived greenhouse gases in our atmosphere indicate that climate change is an issue society will be dealing with for a long time. Climate warming has the potential to affect most aspects of society, including water supplies, agriculture, ecosystems, and economies. NOAA will continue to monitor these gases into the future to further understand the impacts on our planet. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 7:29 a.m. Eastern Time. Quick note. The AGGI is analogous to the dial on the electric blanket. The dial does not tell you exactly how hot you will get, nor does the AGGI predict a specific temperature. Just as turning the dial up increases the heat of an electric blanket, a rise in the AGGI means greater greenhouse warming. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 7:31 a.m. Eastern Time. Quick note. NOAA scientists created the AGGI, recognizing that carbon dioxide is not the only greenhouse gas affecting the balance of heat in the atmosphere. Many other long-lived gases also contribute to warming, although not currently as much as carbon dioxide. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 7:34 a.m. Eastern Time. Quick note. The AGGI includes methane and nitrous oxide, for example, greenhouse gases that are emitted by human activities and also have natural sources and sinks. It also includes several chemicals known to deplete Earth's protective ozone layer, which are also active as greenhouse gases. The 2010 AGGI reflects several changes in the concentration of these gases, including and good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 7:36 a.m. Eastern Time. Quick note. A continued steady increase in carbon dioxide. Global carbon dioxide levels rose to an average of 389 parts per million in 2010, compared with 386 parts per million in 2009 and 354 in the index comparison year of 1990. Before the Industrial Revolution in the 1880s, carbon dioxide concentration in the atmosphere was about 280 parts per million. Carbon dioxide levels swing up and down in natural seasonal cycles, but human activities, primarily the burning of coal, oil, and gas for transportation and power, have driven a consistent upward trend in concentration. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 7:39 a.m. Eastern Time. Quick note. A continued recent increase in methane. Methane levels rose in 2010 for the fourth consecutive year after remaining nearly constant for the preceding 10 years, up to 1799 parts per million. Methane measured 1794 parts per million in 2009 and 1714 parts per billion in 1990. Pound for pound, methane is 25 times more potent as greenhouse gas than carbon dioxide, but there is less of it in the atmosphere. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 7:40 a.m. Eastern Time. Quick note. A continued steady increase in nitrous oxide, best known as laughing gas in dentistry. Nitrous oxide is also a greenhouse gas emitted from natural sources and as a byproduct of agricultural fertilization, livestock, manure, sewage treatment, and some industrial processes. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 7:41 a.m. Eastern Time. Quick note. A continued recent drop in 2-chlorofluorocarbons, CFC11 and CFC12 levels of these two compounds, which are ozone-depleting chemicals in addition to greenhouse gases, have been dropping at about 1% per year since the late 1990s because of an international agreement, Montreal Protocol, to protect the ozone layer. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 7:43 a.m. Eastern Time. Quick note. Scientists at NOAA's Earth System Research Laboratory prepare the AGGI each year from atmospheric data collected through the International Cooperative Air Sampling Network of more than 100 sites around the world. NOAA researchers developed the AGGI in 2004 and so far back calculated it to 1978. Atmospheric composition data from ice core and other records could allow the record to be extended back centuries. And good morning, it's Dr. Will, CBMD, PhD. It is Thursday, November 10, 2011, 7:51 a.m. Eastern Time. Quick note. Physicists chip away at a mystery of antimatter imbalance. Science Daily, November 9, 2011. Why there is stuff in the universe, more properly why there is an imbalance between matter and antimatter, is one of the long-standing mysteries of cosmology. A team of researchers working at the National Institute of Standards and Technology, NIST, has just concluded a 10-year-long study of the fate of neutrons in an attempt to resolve the question, the most sensitive such measurement ever made. The universe, they concede, has managed to keep its secret for the time being, but they've succeeded in significantly narrowing the number of possible answers. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 7:53 a.m. Eastern Time. Quick note. Though the word itself evokes science fiction, antimatter is an ordinary, if highly uncommon, material that cosmologists believe once made up almost exactly half of the substance of the universe. When particles and their antiparticles come into contact, they instantly annihilate one another in a flash of light billions of years ago. Most of the matter and all of the antimatter vanished in this fashion, leaving behind a tiny bit of matter unwashed in cosmic energy. What we see around us today, from stars to rocks to living things, is made up of that excess matter, which survived because a bit more of it existed. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 7:56 a.m. Eastern Time. Quick note. The question is, why was there an excess of one over the other in the first place? A physicist at the NIST Physical Measurement Lab, there are lots of theories attempting to explain the imbalance, but there is no experimental evidence to show that any of them can account for it. It is a huge mystery on the level of asking why the universe is here, except in physics can't explain it. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 7:58 a.m. Eastern Time. Quick note. An answer might be found by examining radioactivity in neutrons, which decay in two different ways that can be distinguished by a specially configured detector. Though all observations thus far have invariably shown these two ways occur with equal frequencies in nature. Finding a slight imbalance between the two would imply that nature favors conditions that would create a bit more matter than antimatter, resulting in the universe we recognize. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 8:01 a.m. Eastern Time. Quick note. Collaborators from several institutions used a detector at the NIST Center for Neutron Research to explore this aspect of neutron decay with greater sensitivity than was ever possible before. For the moment, the larger answer has eluded them. Several years of observation and data analysts once again turned up no imbalance between the two decay paths. But the improved sensitivity of their approach means that they can severely limit some of the numerous theories about the universe antimatter imbalance. And with future improvements to a detector, their approach may help constrain the possibilities far more dramatically. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 8:01 a.m. Eastern Time. Quick note. We have placed very tight constraints on what these theories can say. We have given theories something to work with, and if we can modify our detector successfully, we can envision limiting large classes of theories. It will help ensure the physics community avoids traveling down blind alleys. And good morning, it's Dr. Will, CBMD, PhD. Is Thursday, November 10, 2011, 8:03 a.m. Eastern Time. Quick note. Two types of neutron decay produce a proton, an electron and electron antineutrino, but eject them in different configurations. The experiment in this detected no imbalance, but the improved sensitivity could help place limits on competing theories about the matter-antimatter imbalance in the universe. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 8:07 a.m. Eastern Time. Quick note. No need to shrink guts to have a larger brain. Science Daily, November 9, 2011. The so-called expensive tissue hypothesis, which suggests a trade-off between the size of the brain and the size of the digestive tract, has been challenged by researchers at the University of Zurich. They have shown that brains in mammals have grown over the course of evolution without the digestive organs having to become smaller. The researchers have further demonstrated that the potential to store fat often goes hand in hand with relatively small brains, except in humans who have owed their increased energy intake and correspondingly large brain to communal child care, better diet, and their ability to walk upright. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 8:09 a.m. Eastern Time. Quick note. Brain tissue is a major consumer of energy in the body. If an animal species evolves a larger brain than its ancestors, the increased need for energy can be met by either obtaining additional sources of food or by trade-off with other functions in the body. In humans, the brain is 3 times larger, thus requires a lot more energy than that of our closest relatives, the great apes. Until now, the generally accepted theory for the condition was that early humans were able to redirect energy to their brains thanks to the reduced digestive tract. Zurich primologists, however, have now disproved this theory, demonstrating that mammals with relatively large brains actually tend to have a somewhat bigger digestive tract. The first author of the study, recently published in Nature, has studied hundreds of care cases from zoos and museums. And good morning, it's Dr. Will, CBMD, PhD. This Thursday, November 10, 2011, 8:13 a.m. Eastern Time. Quick note. The data set contains 100 species from the stag to the shrew, explains the PhD student. The scientists involved the study then compared the size of the brain with the fat-free body mass. Senior author Karen Isler stresses that it is extremely important to take an animal's adipose deposits into consideration, as in some species these constitute up to half of the body mass in autumn. But even compared with the fat-free body mass, the size of the brain does not correlate negatively with the mass of other organs. And good morning, it's Dr. Will, CBMD, PhD. Thursday, November 10, 2011, 8:14 a.m. Eastern Time. Quick note. More fat, smaller brain. Nevertheless, the storage of fat plays a key role in the brain size evolution. The researchers discovered another rather surprising correlation. The more fat an animal species can store, the smaller its brain, although adipose tissue itself does not use much energy. Fat animals need a lot more energy to carry extra weight, especially when climbing or running. This energy is then lacking for potential brain expansion. It seems that large adipose deposits often come at the expense of mental flexibility. We humans are an exception, along with whales and seals, probably because, like swimming, our bipedalism doesn't require much more energy, even when we are a bit heavier. And good morning, it's Dr. Will, CBMD, PhD. It's Thursday, November 10, 2011, 8:20 a.m. Eastern Time. Quick note. Interplay of energetic factors. The rapid increase in brain size and the associated increase in energy intake began about 2 million years ago in the genus Homo, based on their extensive studies on animals. Researchers propose a scenario in which several energetic factors are involved. In order to stabilize the brain's energy supply on a higher level, prehistoric man needed an all-year high-quality source of food, such as underground tubers or meat. As they no longer climbed every day, they perfected the art of walking upright. Even more important, however, is communal childcare. Because ape mothers do not receive any help. They can only raise an offspring every 5 to 8 years. Thanks to a communal care for mothers and children, humans can afford both a huge brain and more frequent offspring. And good morning, it's Dr. Will, CBMD, PhD. It's Friday, November 11, 2011, 12:12 a.m. Eastern Time. Quick note. Weird world of water gets a little weirder. Science Daily, November 9, 2011. Strange, stranger, strangest. To the world nature of one of the simplest chemical compounds. The stuff so familiar that even non-scientists know. Its chemical formula add another odd twist. Scientists are reporting that good old H2O, when chilled below freezing point, can shift into a new type of liquid. And good morning, it's Dr. Will, CBMD, PhD. It's Friday, November 11, 2011, 12:15 a.m. Eastern Time. Quick note. Water is one weird substance exhibiting more than 80 unusual properties, by one count including some that scientists still struggle to understand. For example, water can exist in all three states of matter, solid state, liquid, and gas, at the same time, and the forces at its surface enable insects to walk on water, and water to rise up from the roots into the leaves of trees and other plants. And good morning, it's Dr. Will, CBMD, PhD. It's Friday, November 11, 2011, 12:16 a.m. Eastern Time. Quick note. In another strange turn, scientists have proposed that water can go from being one type of liquid into another in so-called liquid-liquid phase transition. But it is impossible to test this with today's laboratory equipment because these things happen so fast. That's why scientists use computer simulations to check it out. And good morning, it's Dr. Will, CBMD, PhD. It's Friday, November 11, 2011, 12:18 a.m. Eastern Time. Quick note. They found that when they chilled liquid water in their simulation, its propensity to conduct heat decreases, as expected for an ordinary liquid. But when they lowered the temperature to about 54 degrees below 0 Fahrenheit, the liquid water started to conduct heat even better in the simulation. Their studies suggest that below this temperature, liquid water undergoes sharp but continuous structural changes, whereas the local structure of liquid becomes extremely ordered. Very much like ice, these structural changes in liquid water lead to increase of heat conduction at lower temperatures. And good morning, it's Dr. Will, CBMD, PhD. It's Friday, November 11, 2011, 12:21 a.m. Eastern Time. Quick note. Giant planets ejected from the solar system. Science Daily, November 10, 2011. Just as an expert chess player sacrificed a piece to protect the queen, the solar system may have given up a giant planet and spared Earth, according to an article recently published in the Astrophysical Journal of What Is. And good morning, it's Dr. Will, CBMD, PhD. It's Friday, November 11, 2011, 12:21 a.m. Eastern Time. Quick note. These clues suggest that the orbits of giant planets were affected by a dynamical instability when the solar system was only about 600 million years old. As a result, the giant planets and smaller bodies scattered away from each other. And good morning, it's Dr. Will, CBMD, PhD. It's Friday, November 11, 2011, 12:23 a.m. Eastern Time. Quick note. Some small bodies moved into the Kepler belt. Others traveled inward, producing impacts on the terrestrial planets and the moon. The giant planets moved as well. Jupiter, for example, scattered most small bodies outward and moved inward. And good morning, this is Dr. Will, CBMD, PhD. It's Friday, November 11, 2011, 12:24 a.m. Eastern Time. Quick note. This scenario presents a problem. However, slow changes in Jupiter's orbit, such as the ones expected from interaction with small bodies, would have conveyed too much momentum to the orbits of the terrestrial planets, stirring up or disrupting the inner solar system and possibly causing Earth to collide with Mars or Venus. And good morning, it's Dr. Will, CBMD, PhD. It's Friday, November 11, 2011, 12:25 a.m. Eastern Time. Quick note. Colleagues suggested a clever way around this problem. They proposed that Jupiter's orbit quickly changed when Jupiter scattered off of Uranus and Neptune during the dynamical instability of the outer solar system. The jumping Jupiter theory, as it is known, is less harmful to the inner solar system because the orbital coupling between the terrestrial planets and Jupiter is weak if Jupiter jumps. And good morning, it's Dr. Will, CBMD, PhD. It is Friday, November 11, 2011, 12:27 a.m. Eastern Time. Quick note. Conducted thousands of computer simulations of the early solar system to test the jumping Jupiter theory. He found that, as hoped for, Jupiter did in fact jump by scattering from Uranus or Neptune. When it jumped, however, Uranus or Neptune was knocked out of the solar system. Something was clearly wrong. And good morning, it's Dr. Will, CBMD, PhD. It is Friday, November 11, 2011, 12:28 a.m. Eastern Time. Quick note. Wondered whether the early solar system could have had 5 giant planets instead of 4 by running the simulations with an additional giant planet with mass similar to that of Uranus and Neptune. Things suddenly fell in place. one planet was ejected from solar system to Jupiter, leaving 4 giant planets behind, and Jupiter jumped, leaving the terrestrial planets undisturbed. And good morning, it's Dr. Will, CBMD, PhD. It's Friday, November 11, 2011, 12:22 a.m. Eastern Time. Quick note. The possibility that the solar system had more than 4 giant planets initially. The ejected sum appears to be conceivable in view of the recent discovery of a large number of free-floating planets in interstellar space, indicating that planet ejection process could be a common occurrence. And good morning, it's Dr. Will, CBMD, PhD. It's Friday, November 11, 2011, 12:37 a.m. Eastern Time. Quick note. Nano car has molecular 4-wheel drive, smallest electric car in the world. Science Daily, November 10, 2011. Reduced to the max, the emission-free, noiseless 4-wheel drive car, jointly developed by EMPA researchers and their Dutch colleagues, represents lightweight construction at its most extreme. The nano car consists of just a single molecule and travels on 4 electrically driven wheels in an almost straight line over a copper surface. The prototype can be admired on the cover of the latest edition of Nature. Good morning, it's Dr. Will, CBMD, PhD. It's Friday, November 11, 2011, 12:43 a.m. Eastern Time. Quick note. To carry out mechanical work, one usually turns to engines, which transform chemical, thermal, or electrical energy into kinetic energy in order to, say, transport goods from A to B. Nature does the same thing in cells called motor proteins, such as chysin and the muscle protein actin, carry out this task. Usually they glide along other proteins similar to a train on rails, and in the process burn ATP, adenosine triphosphate, the chemical fuel, so to speak, of the living world. And good morning, it's Dr. Will, CBMD, PhD. It's Friday, November 11, 2011, 12:56 a.m. Eastern Time. Quick note. A number of chemists aim to use similar principles and concepts to design molecular transport machines, which can then carry out specific tasks on a nanoscale. A decisive step on the road to artificial nanoscale transport systems. They have synthesized a molecule from 4 rotating motor units, i.e. wheels which can travel straight ahead in a controlled manner. To do this, our car needs neither rails nor petrol. It runs on electricity. It must be the smallest electric car in the world, and it even comes with 4-wheel drive, comments EMPA researchers. And good morning, it's Dr. Will, CBMD, PhD. It's Friday, November 11, 2011, 12:58 a.m. Eastern Time. Quick note. Range per tank of fuel, still room for improvement. The downside, small car, which measures approximately 4 times 2 nanometers, about 1 billion times smaller than VW Golf, needs to be refueled with electricity after a half revolution of the wheels, via the tip scanning tunnel microscope, STM. Further, due to the molecular design, the wheels can only turn in one direction. In other words, there's no reverse gear. Good morning, it's Dr. Will, CBMD, PhD. It's Friday, November 11, 2011, 1:02 a.m. Eastern Time. Quick note. Another experiment showed that the molecule really does behave as predicted. A part of the molecule can rotate freely around the central axis, a CC single bond, the chassis of the car, so to speak. It can therefore land on the copper surface in 2 different orientations. In the right one, in which all 4 wheels turn in the same direction, and in the wrong one, in which the rear axle wheels turn forwards but the front ones turn backwards. Upon excitation, the car remains at a standstill. And good morning, it's Dr. Will, CBMD, PhD. It's Friday, November 11, 2011, 1:03 a.m. Eastern Time. Quick note. Therefore, the researchers have achieved the first objective, a proof of concept, i.e., they have been able to demonstrate that individual molecules can absorb external electrical energy and transform it into targeted motion. And good morning, it's Dr. Will, CBMD, PhD. It's Friday, November 11, 2011, 1:07 a.m. Eastern Time. Quick note. Can fetus sense mother's psychological state? Studies suggest yes. Science Daily, November 10, 2011. As a fetus grows, it's constantly getting messages from its mother. It's not just hearing her heartbeat and whatever music she might play in her belly. It also gets chemical signals through a placenta. A new study, which will be published in the Psychological Science, a journal of the Association of Psychological Science, finds that this includes signals about the mother's mental state. If the mother is depressed, that affects how the baby develops after it's born. And good morning, it's Dr. Will, CBMD, PhD. It's Friday, November 11, 2011, 1:09 a.m. Eastern Time. Quick note. In recent decades, researchers have found that the environment a fetus growing up in the mother's womb is very important. Some effects are obvious. Smoking and drinking, for example, can be devastating, but others are subtler. Studies have found that people who were born during the Dutch 1944, most of whom had starving mothers, were likely to have health problems like obesity and diabetes later. And good morning, it's Dr. Will, CBMD, PhD. It is Friday, November 11, 2011, 1:11 a.m. Eastern Time. Quick note. For this study, they recruited pregnant women and checked them for depression before and after they gave birth. They also gave their babies tests after they were born to see how well they were developing. They found something interesting. What mattered to the babies was if the environment was consistent before and after birth. That is, the babies who did the best were those who either had mothers who were healthy both before and after birth, and those whose mothers were depressed before birth and stayed depressed afterward. What slowed the baby's development was changing conditions, a mother who went from depressed before birth to healthy after or healthy before birth, or to depressed after. We must admit the strength of this finding surprised us. And good morning, it's Dr. Will, CBMD, PhD. It is Friday, November 11, 2011, 1:13 a.m. Eastern Time. Quick note. Now, the cynical interpretation of our results would be that if a mother is depressed before birth, you should leave her that way for the well-being of the infant. A more reasonable approach would be to treat women who present with prenatal depression. We know how to deal with depression. The problem is women are rarely screened for depression before birth. And good morning, it's Dr. Will, CBMD, PhD. It is Friday, November 11, 2011, 1:14 a.m. Eastern Time. Quick note. In the long term, having a depressed mother could lead to neurological problems and psychiatric disorders. In another study, his team found that older children whose mothers were anxious during pregnancy, which often is comorbid with depression, having differences in certain brain structures. It will take studies lasting decades to figure out exactly what happened a depressed mother means to a child's long-term health. And good morning, it's Dr. Will, CBMD, PhD. It is Friday, November 11, 2011, 2:04 a.m. Eastern Time. Quick note. Chemists reveal the force within you. New method for visualizing mechanical forces on cell surface. Science Daily, November 9, 2011. A new method for visualizing mechanical forces on the surface of a cell, reported in Nature Methods, provides the first detailed view of those forces as they occur in real time. And good morning, it's Dr. Will, CBMD, PhD. It is Friday, November 11, 2011, 2:05 a.m. Eastern Time. Quick note. Now we are able to measure something that's never been measured before. The force that one molecule applies to another molecule across the entire surface of a living cell. And as this cell moves and goes about its normal processes. And good morning, it's Dr. Will, CBMD, PhD. It is Friday, November 11, 2011, 2:07 a.m. Eastern Time. Quick note. Cells are constantly tugging and pushing on their surroundings, and they can even communicate with one another using mechanics. One way that cells use forces is evident from the characteristic architecture of tissue, like a lung or a heart. If we want to really understand cells and how they work, we have to understand cell mechanics at a molecular level. The first step is to measure the tension applied to specific receptors on the cell surface. And good morning, it's Dr. Will, CBMD, PhD. It's Friday, November 11, 2011, 2:08 a.m. Eastern Time. Quick note. The researchers demonstrated their technique on the epidermal growth factor receptor, EGFR, one of the most studied cellular signaling pathways. They mapped the mechanical strain exerted by EGFR during the early stages of endocytosis. When the protein receptor of the cell takes in a ligand, or a binding molecule, the result showed that the cell does not previously absorb the ligand but physically pulls it inside during the process. Their experiments provide the first direct evidence that force is exerted during endocytosis. And good morning, it's Dr. Will, CBMD, PhD. It is Friday, November 11, 2011, 2:10 a.m. Eastern Time. Quick note. It is known that if EGFR is overactive, that can lead to cancer. And one of the ways that EGFR is activated is by binding its ligand and taking it on. So if we can understand how tugging on EGFR forces changes the pathway and whether it plays a role in cancer, it might be possible to design drugs that target this pulling process. And good morning, it's Dr. Will, CBMD, PhD. It is Friday, November 11, 2011, 2:11 a.m. Eastern Time. Quick note. Several methods have been developed in recent years to try to study the mechanics of cellular forces, but they have major limitations. One genetic engineering approach requires splitting open and modifying protein of a cell. This invasive technique may change the behavior of the cell, skewing the results. And good morning, it's Dr. Will, CBMD, PhD. It is Friday, November 11, 2011, 2:13 a.m. Eastern Time. Quick note. The technique developed is non-invasive, does not modify the cell, and can be done with a standard fluorescent microscope. A flexible polymer is chemically modified at both ends. One end in a fluorescent base turn on sensor that will bind to a receptor on the cell surface. The other end is chemically anchored to a microscope slide and a molecule that quenches fluorescence. And good morning, it's Dr. Will, CBMD, PhD. It is Friday, November 11, 2011, 2:13 a.m. Eastern Time. Quick note. Once a force is applied to the polymer, it stretches out. The distance from the quencher increases and the fluorescent signal turns on and grows brighter. We can determine the force being exerted by measuring the amount of fluorescent light emitted. And good morning, it's Dr. Will, CBMD, PhD. It is Friday, November 11, 2011, 2:14 a.m. Eastern Time. Quick note. The forces of any individual protein or molecule on the cell surface can be measured using the technique at a far higher spatial or temporal resolution than was previously possible. And good morning, it's Dr. Will, CBMD, PhD. This Friday, November 11, 2011, 2:16 a.m. Eastern Time. Quick note: many mysteries beyond the biology and chemistry of cells may be explained through measuring cellular forces. How does a cancer cell crawl when a tumor spreads? What are the forces involved in the cell division and immune response? What are the mechanics that allow groups of cardiac cells to beat in unison? And good morning, it's Dr. Will, CBMD, PhD. This Friday, November 11, 2011, 2:17 a.m. Eastern Time. Quick note: our method can be applied to nearly any receptor, opening the door to rapidly studying chemical and mechanical interactions across the thousands of membrane-bound receptors on the surface of virtually any cell type. We hope that measuring cellular forces could then become part of a standard repertoire of biochemical techniques that scientists use to study living systems. And good morning, it's Dr. Will, CBMD, PhD. This Friday, November 11, 2011, 2:22 a.m. Eastern Time. Quick note: sea change can forest South America wildfire. Science Daily, November 10, 2011. Tiny temperature changes on Atlantic and Pacific Oceans provide an excellent way to forecast wildfires in South America rainforest. And good morning, it's Dr. Will, CBMD, PhD. This Friday, November 11, 2011, 2:23 a.m. Eastern Time. Quick note: the prediction method is similar to that for storms. Just as sea surface temperatures can be harbingers of powerful Atlantic hurricanes, El Nino conditions that influence rainfall in California and elsewhere on the Pacific Rim, they can be used to model drought-inducing atmospheric changes utilizing archival satellite data from NASA. Ken and fellow scientists painstakingly plotted a decade of water temperature variation in wildfire duration and intensity across Central South America. And good morning, it's Dr. Will, CBMD, PhD. This Friday, November 11, 2011, 2:25 a.m. Eastern Time. Quick note: they found that temperatures increase of as little as a quarter of a degree Celsius on the Atlantic and a single degree on the Pacific, matched up with more deadly blazes across broad swaths of the Amazon over the next year. These changes are slight, but they trigger big effects in these tropical forest ecosystems. Ocean temperatures affect atmospheric winds and clouds that can reduce or increase rainfall over the continent. And good morning, it's Dr. Will, CBMD, PhD. This Friday, November 11, 2011, 2:27 a.m. Eastern Time. Quick note: the scientists tested their theory by forecasting based on water temperature readings, South America 2010 fire season, and then checking actual data afterward. Their accuracy was striking. Last year saw the biggest temperature increase on both oceans, and several months later, far drier conditions on land. And good morning, it's Dr. Will, CBMD, PhD. It's Friday, November 11, 2011, 2:29 a.m. Eastern Time. Quick note: we predicted a massive spike in fires in 2010, and it occurred, citing conflagrations across hundreds of miles in one state. They had forest worse fires than occurred. Then learned later that officials there had sharply reduced clear-cutting the year before. Overall, while deforestation has declined in the past decade, wildfires have not, because the prescribed farming burns that escape into nearby woods. And good morning, it's Dr. Will, CBMD, PhD. It's Friday, November 11, 2011, 2:30 a.m. Eastern Time. Quick note: the scientists are compiling data for 2012 fire season and plan to release their findings this winter for officials to use next year. The next step will be to see if the prediction method works for forest in Siberia, Indonesia, and West Africa. And good morning, it's Dr. Will, CBMD, PhD. This is Monday, November 7, 2011, 6:15 a.m. Eastern Time. Quick note: scientists find evidence of ancient mega drought in southwestern U.S. Science Daily, November 6, 2011. A new study at the University of Arizona Laboratory of Tree Ring Research has revealed a previously unknown multi-decade drought period in the 2nd century A.D. The findings give evidence that extended periods of aridity have occurred at intervals throughout our past. And good morning, it's Dr. Will, CBMD, PhD. This is Monday, November 7, 2011, 6:16 a.m. Eastern Time. Quick note: almost 900 years ago, in the mid-12th century, the southwestern U.S. was in the middle of a multi-decade mega drought. It was the most recent extended period of severe drought known for this region, but it was not the first. The 2nd century A.D. saw an extended dry period of more than 100 years, characterized by the multi-decade drought lasting nearly 50 years, says a new study from the scientists at the University of Arizona. And good morning, it's Dr. Will, CBMD, PhD. This is Monday, November 7, 2011, 6:20 a.m. Eastern Time. Quick note: dendrochronology is a precise science of using annual growth rings of trees to understand climate in the past. Because trees add a normally clearly defined growth ring around their trunk each year, counting the rings backwards from the tree's bark allows scientists to determine not only the age of the tree, but which years were good for growth and which years were more difficult. And good morning, it's Dr. Will, CBMD, PhD. This is Monday, November 7, 2011, 6:20 a.m. Eastern Time. Quick note: if it is a wet year, they grow a wide ring, and if it is a dry year, they grow a narrow ring. If you average that pattern across trees in the region, you can develop a chronology that shows what year were drier or wetter for that particular region. And good morning, it's Dr. Will, CBMD, PhD. It is Monday, November 7, 2011, 6:21 a.m. Eastern Time. Quick note. Darker wood referred to as late wood because it develops in the later part of the year at the end of the growing season. Forms a usually distinct boundary between one ring and the next. The late wood is darker because growth at the end of the growing season has slowed, and the cells are more compact. And good morning, it's Dr. Will, CBMD, PhD. It is Monday, November 7, 2011, 6:23 a.m. Eastern Time. Quick note. Literally nothing is older than brisk-cone pine tree, the oldest and longest living species on the planet. These pine trees normally are found clinging to bare rock landscapes of alpine or near alpine mountain slopes. The trees, the oldest of which are more than 4,000 years old, are capable of withstanding extreme drought conditions. And good morning, it's Dr. Will, CBMD, PhD. It is Monday, November 7, 2011, 6:24 a.m. Eastern Time. Quick note. To sample the trees without damaging them, the dendrochronologists use a tool like a metal screw that bores a tiny hole in the trunk of the tree and allows them to extract a sample called a core. We take a piece of wood about the size and shape of a pencil from a tree. And good morning, it's Dr. Will, CBMD, PhD. It is Monday, November 7, 2011, 6:25 a.m. Eastern Time. Quick note. We also sampled dead wood that was lying about the land. We took our samples back to the lab where we used a visual graphic technique to match where the annual growth patterns of the living trees overlap with the patterns in the dead wood. Once we have the pattern matched, we measure the rings and average these values to generate a site chronology. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 6:26 a.m. Eastern Time. Quick note. In our chronology for the South San Juan Mountains, we created a record that extends back to 2,200 years. It was pretty profound that we were able to get back that far. The chronology extends many years earlier than the medieval period, during which two major drought events in that region already were known from previous chronologies. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 6:27 a.m. Eastern Time. Quick note. The medieval period extends roughly from 800 to 1300 AD. During that period, there was a lot of evidence from previous studies for increased aridity, in particular two major droughts, one in the middle of the 12th century and one at the end of the 13th century. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 6:28 a.m. Eastern Time. Quick note. Very few records are long enough to assess the global conditions associated with these two periods of southwestern aridity, and the available records have uncertainties. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 6:29 a.m. Eastern Time. Quick note. There was another period of increased aridity even earlier. This new record shows that in addition to known droughts from the medieval period, there is also evidence for an earlier mega-drought during the 2nd century AD. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 6:30 a.m. Eastern Time. Quick note. What we can see from our record is that it was a period of basically 50 consecutive years of below average growth. And that's within a much broader period that extends from around 124 AD to 210 AD, about a 100-year-long period of dry conditions. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 6:31 a.m. Eastern Time. Quick note. We are showing that there are multiple extreme drought events that happened during our past in this region. These mega droughts lasted for decades, which is much longer than our current drought, and the climatic events behind these previous dry periods are really similar to what we are experiencing today. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 6:33 a.m. Eastern Time. Quick note. The prolonged drought in the 12th century and the newly discovered event in the 2nd century AD may have been influenced by a warmer than average northern hemisphere temperature. The limited records indicated there may have been similar La Nina-like background conditions in the tropical Pacific Ocean, which are known to influence modern drought during the two periods. And good morning, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 6:35 a.m. Eastern Time. Quick note. Although natural climatic variation has led to an extended dry period in the southwestern U.S., in the past there is a reason to believe that the human-driven climatic change will increase the frequency of extreme droughts in the future. In other words, we should expect similar multi-decade droughts in the future, predicted to be even warmer than the past. And good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 5:50 p.m. Eastern Time. Quick note. Erasing signs of aging in human cells, now a reality. Science Daily, November 3, 2011. Scientists have recently succeeded in rejuvenating cells from elderly donors aged over 100 These old cells were reprogrammed in vitro to induce pluripotent stem cells, iPSC, and two rejuvenated and human embryonic stem cells, HESC. Cells of all types can again be differentiated after this genuine rejuvenation therapy. The results represent significant progress for research into iPSC cells and a further step forward for regenerative medicine. And good morning, it's Dr. Will, CBMD, PhD. It's Monday, November 7, 2011, 5:53 p.m. Eastern Time. Quick note. Human embryonic stem cells, HESC, are undifferentiated multiple function cells. They can divide and form all types of differentiated adult cells in the body, neurons, cardiac cells, skin cells, liver cells, etc. Since 2007, a handful of research teams across the world have been capable of reprogramming human adult cells into induced pluripotent cells, iPSC, which have similar characteristics and potential to human embryonic stem cells, HESC. This kind of reprogramming makes it possible to reform all human cell types without the ethical restrictions related to using embryonic stem cells. And good evening, it's Dr. Will, CBMD, PhD. It is Monday, November 7, 2011, 5:56 p.m. Eastern Time. Quick note. Until now, research results demonstrated that since the final stage of cellular aging was an obstacle blocking the use of the technique for therapeutical applications in elderly patients. And his team have overcome this obstacle. The researchers have successfully rejuvenated cells from elderly donors, some over 100 years old, thus demonstrating the reversibility of the cellular aging process. And good evening, it's Dr. Will, CBMD, PhD. It is Monday, November 7, 2011, 5:58 p.m. Eastern Time. Quick note. To achieve this, they used an adapted strategy that consisted of reprogramming cells using a specific cocktail of 6 genetic factors while erasing signs of aging. The researchers provided that the iPSC cells thus obtained then had the capacity to reform all types of human cells. They have the physiological characteristics of young cells, both from the perspective of their proliferative capacity and their cellular metabolisms. And good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 6:01 p.m. Eastern Time. Quick note. Using this new cocktail of 6 factors, the senescent cells reprogrammed into functional iPSC cells required the characteristic of embryonic pluripotent stem cells. Good evening, it's Dr. Will, CBMD, PhD. It's Monday, November 7, 2011, 6:05 p.m. Eastern Time. Quick note. In particular, they recovered their capacity for self-renewal and their form or differentiation potential. They do not preserve any traces of previous aging. To check the rejuvenated characteristics of these cells, the researchers tested the reverse process. The rejuvenated iPSC cells were again differentiated to adult cells and compared to the original old cells, as well as to those obtained using human embryonic pluripotent stem cells, HESC. And good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 6:08 p.m. Eastern Time. Quick now. Signs of aging were erased, and the iPSC cells obtained can produce functional cells of any type with an increased proliferation capacity of longevity. And good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 6:09 p.m. Eastern Time. Quick note. Tested on the cells taken from donors over the age of 100. The results obtained led to the research team to test the cocktail on even older cells taken from donors of 92, 94, 96, and even up to 101 years old. Our strategy worked on cells taken from donors in their hundreds. The age of cells is definitely not a reprogramming barrier. This research paves the way for their therapeutical use of IPS, insofar as an ideal source of adult cells is provided, which are tolerated by the immune system and repair organs or tissues in elderly patients. And good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 6:56 p.m. Eastern Time. Quick note. Brain parasite directly alters brain chemistry. Science Daily, November 4, 2011. Research shows infection by the brain parasite Toxoplasma gondii, found in 10 to 20% of the UK's population, directly affects the production of dopamine, a key chemical messenger in the brain. And good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 6:57 p.m. Eastern Time. Quick note. Faculty of Biological Sciences believes that the findings could ultimately shed new light on treating human neurological disorders that are dopamine-related, such as schizophrenia, attention deficit hyperactivity disorder, and Parkinson's disease. And good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 6:58 p.m. Eastern Time. Quick note. This research may explain how these parasites remarkably manipulate rodents, behavioral for their own advantage. Infected mice and rats lose their innate fear of cats, increasing the chances of being caught and eaten, which enables the parasite to return to its main host to complete its life cycle. And good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 6:59 p.m. Eastern Time. Quick note. Stanley Medical Research Institute and Dunhill Medical Trust. The research team found that the parasite causes production and release of many times the normal amount of dopamine in infected brain cells. And good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 7:01 p.m. Eastern Time. Quick note. Dopamine is a natural chemical which relays messages in the brain-controlling aspects of movement, cognition, and behavior. It helps control the brain's reward and pleasure centers and regulates emotional responses such as fear. The presence of a certain kind of dopamine receptor is also associated with sensation seeking, whereas dopamine deficiency in humans results in Parkinson's disease. And good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 7:01 p.m. Eastern Time. Quick note. Based on these analyses, it was clear that T. gondii can orchestrate a significant increase in dopamine production in neural cells. And good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 7:06 p.m. Eastern Time. Quick note. Humans are accidental hosts to T. gondii, and the parasite could end up anywhere in the brain. So human symptoms of toxoplasmosis infection may depend on where a parasite ends up. This may explain the observed statistical link between incidences of schizophrenia and toxoplasmosis infection. And good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 7:07 p.m. Eastern Time. Quick note. Next experiments will investigate how the parasite enzyme triggers dopamine production and how this may change behavior. And good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 7:08 p.m. Eastern Time. Quick note. Toxoplasmosis, which is transmitted via cat feces found on unwashed vegetables and raw or undercooked infected meat, is relatively common, with 10 to 20% of UK population and 22% of the US population estimated to carry the parasite as cyst. Most people with the parasite are healthy, but for those who are immune suppressed, and particularly for pregnant women, there are significant health risks that can occasionally be fatal. And good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 7:12 p.m. Eastern Time. Quick note. The parasite infects the brain by forming a cyst within its cells and produces an enzyme called tyrosine hydroxylase, which is needed to make dopamine. Dopamine's role in mood, sociability, attention, motivation, and sleep patterns are well documented, and schizophrenia has long been associated with dopamine, which is the target of all current schizophrenia drugs on the market. And good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 7:13 p.m. Eastern Time. Quick note. The enzyme tyrosine hydroxylase is a crucial step in making L-DOPA, prescribed as levodopa for Parkinson's disease, a chemical that is readily converted to the neurotransmitter dopamine. And good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 7:17 p.m. Eastern Time. Quick note. Tropical forests fertilized by nitrogen air pollution, scientists find. Science Daily, November 3, 2011, scientists braved ticks and a tiger to discover how human activities have perturbed the nitrogen cycle in tropical forests. Studies at the remote Smithsonian Institute Global Earth Observatory sites in Panama and Thailand show the first evidence of long-term effects of nitrogen pollution in tropical trees. And good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 7:23 p.m. Eastern Time. Quick note. Air pollution is fertilizing tropical forests with one of the most important nutrients for growth. Tropical Research Institute in Panama. We compared nitrogen in leaves from dried specimens collected in 1968 with nitrogen in samples of new leaves collected in 2007. Leaf nitrogen concentration in the proportion of heavy to light nitrogen isotopes increased in the last 40 years, just as they did in another experiment when we applied fertilizer to the forest floor. And good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 7:26 p.m. Eastern Time. Quick note. Nitrogen is an element created in stars under high temperatures and pressures. Under normal conditions, it is a colorless, odorless gas that does not readily react with other substances. Air consists of more than 75% nitrogen, but nitrogen also plays a big role in life as an essential component of proteins. When nitrogen gas is zapped by lightning or absorbed by soil bacteria called nitrogen fixers, it is converted into other active forms that can be used by animals and plants. Humans fix nitrogen by the Haber process, which converts nitrogen gas into ammonia, now a principal ingredient in fertilizers. Today, nitrogen fixation by humans has approximately doubled the amount of reactive nitrogen emitted. And good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 7:30 p.m. Eastern Time. Quick note. Nitrogen comes in two forms, or isotopes, atoms that have the same number of protons but different numbers of neutrons. In the case of nitrogen, the isotopes are 14N and 15N, although only about 1 in 300 nitrogen atoms is the heavier form. Imagine nitrogen in the ecosystem like a bowl of popcorn. Normally the ratio of popped light to unpopped heavy kernels stays the same, but when someone starts to eat the popcorn, the lighter the popped kernels get used up first, increasing the ratio of heavy to light kernels, or 15N divided by 14N in the case of the ecosystem. Light nitrogen is lost through nitrate leaching and as gases such as N2 and various forms of nitrous oxides or noxides, some of which can be important greenhouse gases. In the fertilization study in Panama mentioned earlier, N2O emissions were tripled. And good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 7:36 p.m. Eastern Time. Quick note. Tree rings provide a handy timeline for measuring changes in wood nitrogen content. From the Institute of Botany at the University of Natural Resources and Life Science, who faced down a tiger when sampling trees in the monsoon forest on the Thailand border. We find that over the last century there is an increase in the heavier form of nitrogen over the lighter form, which tells us that there is more nitrogen going into the system and higher losses. We also got the same result in an earlier study of tree rings in Brazilian rainforest. So it looks like nitrogen fixed by humans now affects some of the most remote areas in the world. And good evening, it's Dr. Will, CBMD, PhD. It's Monday, November 7, 2011, 7:39 p.m. Eastern Time. Quick note. The results have a number of important implications. The most obvious is for trees in the bean family, Fabaceae, a major group in tropical forests that fix their own nitrogen association with soil bacteria. Increased nitrogen from outside could take away their competitive advantage and make them less common, changing the composition of tree communities. And good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 7:41 p.m. Eastern Time. Quick note. There are also implications for global change models, which are beginning to include nitrogen availability as a factor, affecting the response of plants to increasing atmospheric carbon dioxide concentrations. Most models assume that higher nitrogen equals more plant growth, which could remove carbon from the atmosphere and offset future warming. However, a challenge for the models is that there is no evidence that trees are growing faster in Panama, despite the long-term increases in nitrogen deposition and atmospheric carbon dioxide. Good evening, it's Dr. Will, CBMD, PhD. This Monday, November 7, 2011, 7:43 p.m. Eastern Time. Quick note. Decades of atmospheric nitrogen deposition have caused major changes in the plants and soils of temperate forests in the U.S. and Europe. Whether tropical forest will face similar consequences is an important question for future research. And good morning, it's Dr. Will, CBMD, PhD. This Tuesday, November 8, 2011, 8:45 a.m. Eastern Time. Quick note. The success of the project to create a reprogrammable cell could revolutionize synthetic biology and would pave the way for scientists to create completely new and useful forms of life using a relatively hassle-free approach. And good morning, it's Dr. Will, CBMD, PhD. This Tuesday, November 8, 2011, 8:46 a.m. Eastern Time. Quick note. We are looking at creating a cell's equivalent to a computer operating system in such a way that a given group of cells could be seamlessly reprogrammed to perform any function without needing to modify its hardware. And good morning, it's Dr. Will, CBMD, PhD. This Tuesday, November 8, 2011, 8:47 a.m. Eastern Time. Quick note. We are talking about a highly ambitious goal leading to a fundamental breakthrough that will ultimately allow us to rapidly prototype, implement, and deploy living entities that are completely new and do not appear in nature, adapting them so they perform new useful functions. And good morning, it's Dr. Will, CBMD, PhD. This Tuesday, November 8, 2011, 10:23 p.m. Eastern Time. Quick note. Sea life must swim faster to survive. To survive climate change. Science Daily, November 7, 2011. Fish and other sea creatures will have to travel large distances to survive climate change, international marine scientists have warned. And good evening, it's Dr. Will, CBMD, PhD. This Tuesday, November 8, 2011, 8:23 p.m. Eastern Time. Quick note. Sea life, particularly in the Indian Ocean, the Western and Eastern Pacific, and the subarctic oceans, will face growing pressures to adapt or relocate to escape extinction, according to a new study by an international team of scientists published in the Journal of Science. And good evening, it's Dr. Will, CBMD, PhD. This Tuesday, November 8, 2011, 8:25 p.m. Eastern Time. Quick note. Our research shows that species which cannot adapt to the increasingly warm waters they will encounter under climate change will have to swim farther and faster to find a new home. And good evening, it's Dr. Will, CBMD, PhD. It's Tuesday, November 8, 2011, 10:25 p.m. Eastern Time. Quick note. Using 50 years data of global temperature changes since the 1960s, the researchers analyzed the shifting climates and seasonal patterns on land and in the oceans to understand how this will affect life in both over the coming century. And Good evening, it's Dr. Will, CBMD, PhD. It's Tuesday, November 8, 2011, 10:26 p.m. Eastern Time. Quick note. We examine the velocity of climate change, the geographical shifts of temperature bands over time, and the shift in seasonal temperatures for both land and sea. We found both measures were higher for the ocean at certain latitudes than on land, despite the fact that the oceans tend to warm more slowly than air over the land. And good evening, it's Dr. Will, CBMD, PhD. It's Tuesday, November 8, 2011, 10:27 p.m. Eastern Time. Quick note. The finding has serious implications, especially for marine biodiversity hotspots, such as the famous Coral Triangle and reefs that flourish in equilateral seas, and for life in polar seas, which will come under rising pressure from other species moving in. And good evening, it's Dr. Will, CBMD, PhD. It is Tuesday, November 8, 2011, 10:28 p.m. Eastern Time. Quick note. Unlike land-dwelling animals, which can just move up a mountain to find a cooler place to live, a sea creature may have to migrate several hundred kilometers to find a new home where the water temperature, seasonal conditions, and food supply all suit it. And good evening, it's Dr. Will, CBMD, PhD. It's Tuesday, November 8, 2011, 10:28 p.m. Eastern Time. Quick note. Under current global warming, land animals and plants are migrating polewards at a rate of about 6 kilometers a decade, but sea creatures may have to move several times faster to keep in touch with the water temperature and conditions that best suit them. And good evening, it's Dr. Will, CBMD, PhD. It's Tuesday, November 8, 2011, 10:30 p.m. Eastern Time. Quick note. We have been underestimating the likely impact of climate change on the oceans. As a general rule, it seems sea life will have to move a lot faster and farther to keep up with temperature shifts in the oceans. This applies especially to fish and marine animals living in the equilateral and subarctic seas, and poses a particular issue both for conservation and fishery management. There is also a complex mosaic of responses globally related to local warming and cooling. For example, our analysts suggest that life in many areas in the Southern Ocean could move northward. However, as a rule, they are likely to be as great or greater in the sea than on land, as a result of its more uniform temperature distribution. And good evening, it's Dr. Will, CBMD, PhD. It's Tuesday, November 8, 2011, 10:33 p.m. Eastern Time. Quick note. The migration is likely to be particularly pronounced among marine species living at or near the sea surface, or subsisting on marine plants and plankton that require sunlight, and less so in the deep oceans. And good evening, it's Dr. Will, CBMD, PhD. It's Tuesday, November 8, 2011, 10:33 p.m. Eastern Time. Quick note. Also, as seas around the equator warm more quickly and sea life migrates away north or south in search of cooler water, it isn't clear that, if anything, will replace it. No communities of organisms from even warmer regions currently exist to replace those moving out. And good evening, it's Dr. Will, CBMD, PhD. It's Tuesday, November 8, 2011, 10:34 p.m. Eastern Time. Quick note. At the same time, sea life living close to the poles could find itself overwhelmed by marine migrants moving in from warmer regions in search of cooler water. And good evening, it's Dr. Will, CBMD, PhD. It's Tuesday, November 8, 2011, 10:37 p.m. Eastern Time. Quick note. Castles in the Desert satellites reveal lost cities in Libya. Science Daily, November 7, 2011. Satellite imagery has uncovered new evidence of a lost civilization of Sahara, Libya's southwestern desert waste that will help rewrite the history of the country. The fall of Gaddafi has opened the way for archaeologists to explore the country pre-Islamic heritage so long ignored under his regime. And good evening, it's Dr. Will, CBMD, PhD. It's Tuesday, November 8, 2011, 10:40 p.m. Eastern Time. Quick note. New role for hormone of love, oxytocin in the brain. Helps direct development of brain-body interface. Science Daily, November 2nd, 2011. Much of the body's chemistry is controlled by the brain, from blood pressure to appetite to food metabolism. Structure of one crucial brain area in which biochemical commands are passed from the brain cells to the bloodstream and from there to the body. In the process, the discovery a surprising new role for the hormone of love, showing that it helps to direct the development of this brain structure. And good evening, it's Dr. Will, CBMD, PhD. It's Tuesday, November 8, 2011, 10:42 p.m. Eastern Time. Quick note. The area in question, the neurohypophysis, is an interface between nerve fibers and blood vessel located at the base of the brain. Here, some of the major brain-body interactions take place. Hormones released from nerves into the blood vessels regulate a series of vital body processes, including the balance of fluids and uterine contractions in childbirth. And good evening, it's Dr. Will, CBMD, PhD. It's Tuesday, November 8, 2011, 10:43 p.m. Eastern Time. Quick note. Although the neurohypophysis has been studied for more than a century, the scientists in the Weizmann Institute led the study developed new genetic tools that enable them to examine the exact three-dimensional arrangement of this brain structure and clarify the cellular and molecular processes leading to its formation. Since the human neurohypophysis is exceedingly complex, the scientists performed the research on live embryos of zebrafish. These fully transparent embryos offer a unique model for studying the vertebrate brain, leading themselves to genetic manipulation with relative ease, enabling researchers to observe the actual formation of a neurohypophysis under a microscope. And good evening, it's Dr. Will, CBMD, PhD. It's Tuesday, November 8, 2011, 10:44 p.m. Eastern Time. Quick note: the study revealed a surprising new function for the hormonal messenger oxytocin, dubbed hormone of love because, in addition to controlling appetite, such female reproductive behaviors and breastfeeding, it is also involved mother-child and mate bonding. The scientists showed that oxytocin, one of the two major hormones secreted in the adult neurohypophysis, is involved in the development of the brain area already in the embryo. At this stage, the oxytocin governs the formation of new blood vessels. The messenger helps to build the road for transmitting its own future messages. And good evening, it's Dr. Will, CBMD, PhD. It's Tuesday, November 8, 2011, 10:46 p.m. Eastern Time. Quick note: these findings provide an important advance in basic research because they shed light on the fundamental brain processes, but in the future they might also be relevant to the treatment of disease. Since the neurohypophysis is one of the only few portions of the brain able to regenerate after injury, an understanding of how it is formed may one day help achieve such regeneration in other parts of the central nervous system. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 12:15 a.m. Eastern Time. Quick note: farm fish-fed vegetable matter may have residual pesticides. Science Daily, November 8, 2011. Today, half of all the fish we eat comes from fish farms. The problem is that these fish are increasingly being fed vegetable matter, which could lead to a buildup of residual pesticides in them. A new test shows how high the risk of contamination really is. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 12:17 a.m. Eastern Time. Quick note: people are eating more and more fish. To meet rising demand, fish are increasingly being cultivated in fish farms. Aquaculture is the fastest developing branch of the global food industry, with annual growth of 9%. This rapid rise is also pushing up demand for fish feed producers, of which are facing a formidable challenge. Because the supply of fish meal and fish oil, important ingredients in feed, is dwindling, they are set to be replaced by crops such as soya, maize, and rape. But the trouble with these is that feed pellets made from them might contain pesticides. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 12:19 a.m. Eastern Time. Quick note: to protect consumers, there has to be a way to test fish bred in captivity for pesticide residues, but this has not been possible up till now. While techniques known as the metabolism studies are already used to test how the active ingredients in pesticides accumulate and break down in ruminants, poultry, and pigs, these techniques are not suitable for use with fish. Germany's Federal Office Protection and Food Safety has let it be known that urgent action is required. Now scientists at the Institute of Molecular Biology and Applied Ecology have developed a system to test whether chemical substances accumulate in fish that are fed contaminated feed. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 12:20 a.m. Eastern Time. Quick note: first, we test whether ingestion of the food feeds leads to a buildup of pesticide residues in fish tissue. Then we look to see which degeneration products or metabolites results from the fish metabolite processes. Essentially, the more fat-soluble a substance is, the higher the probability of it accumulating in fish. Our test from the basis for later studies on feeding. The results determine whether these subsequent studies, which ascertain maximum pesticide residue levels, are required. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 12:25 a.m. Eastern Time. Quick note: for their metabolism studies, the researchers use water tanks that are 2 cubic meters in size. Into these tanks, they place carp and rainbow trout, each weighing 300 to 500 grams. Both these freshwater fish are frequently bred in farms to detect and identify pesticide residues in their metabolites. A powerful filtering system prevents the dissolved test substance from accumulating in the water. The researchers then test the fresh of these fish for pesticide residues using highly sensitive analytical methods which permit even the smallest quantities of a substance to be detected with certainty. Our new test leaves no stone unturned in the search for pesticides and their degradation products in fish, from breeding through the tissue analysts in the laboratory. And Good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 12:27 a.m. Eastern Time. Quick note. In autumn 2011, the European Commission will publish new data requirements for fish as part of the approval process for pesticides. These will oblige every producer and importer who intends to bring a new pesticide onto the European market not only to register it, but also to provide information proving it cannot accumulate in the edible parts of fish. The fish test developed at the IME will supply the information required. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 12:38 a.m. Eastern Time. Quick note. 1. You are keenly interested in studying the metabolism of aromatic amino acids in Escherichia coli, particularly the synthesis of phenylalanine. To initiate your investigation, you decide to isolate phenylalanine oxotrophs by exposing wild-type E. coli to a mutagen. An oxotroph is a mutant that cannot grow on media lacking certain essential molecules that are normally synthesized by the wild-type strain. Hence, in this case, phenylalanine oxotroph would require phenylalanine to be present in the medium in order to grow. A mutagen is a chemical or physical agent that interacts with DNA and causes a mutagen to occur. And good morning, it's Dr. Will, CBMD, PhD. This Wednesday, November 9, 2011, 12:50 a.m. Eastern Time. Quick note. Equations and concepts. The cross product of any two vectors, A and B, is a vector C whose magnitude is given by AB sin degrees and whose direction is perpendicular to the plane formed by A and B. The sense of C can be determined from the right-hand rule. C equals A times B, C in bars equals AB sin degrees. End. Good morning, it's Dr. Will, CBMD, PhD. It is Wednesday, November 9, 2011, 12:51 a.m. Eastern Time. Quick note. The angular momentum of a particle whose linear momentum is p and whose vector position is r is defined as L r times p. The SI unit of an angular momentum is kg times m divided by s to the power 2. Note that both the magnitude and direction of L depends on the choice of origin. L r times p. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 12:52 a.m. Eastern Time. Quick note. The torque acting on a particle whose vector position is r can be expressed as r times F, where F is the external force acting on a particle. Torque also depends on the choice of the origin and has an SI unit of N times M. T equals R times F equals N. Good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 12:54 a.m. Eastern Time. Quick note. If the same origin is used to define L and T, then the torque on the particle equals the time rate of change of its angular momentum. This expression is the rotational analog Newton's second law, F equals dp divided by dt, and its basic equation for treating rotating rigid bodies and rotating particles, T equals dl divided by dt. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 12:56 a.m. Eastern Time. Quick note. The angular momentum of a system of particles is obtained by taking the vector sum of the individual angular momenta about some point in the inertial frame. The individual momenta may change with time, which can change the total angular momentum. However, the total angular momentum of a system will only change if a net external torque acts on the system. In fact, the net torque acting on a system of particles equals the time rate of a change of total angular momentum. Infinity T ext equals dl divided by dt. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 12:57 a.m. Eastern Time. Quick note. The angular momentum of a rigid body in the form of a plane lamina rotating about a fixed axis is given by the product IW, where I is the momentum of inertia. About the axis of rotation, W is the angular velocity. L equals IW. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 12:59 a.m. Eastern Time. Quick note. Taking the time derivative and using, we find that the net external torque acting on a rigid body is proportional to the angular acceleration alpha. Hence, if the external torque is 0, the body has no angular acceleration. In this case, the body either rotates with constant angular velocity or is at rest. Infinity T ext equals IA alpha. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 1:01 a.m. Eastern Time. Quick note. The law of conservation of angular momentum states that if the resultant external torque acting on a system is 0, the total angular momentum is constant. This follows. If infinity T ext equals dl divided by dt equals 0, L is constant. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 1:03 a.m. Eastern Time. Quick note. If we apply this result to a body rotating about a fixed axis and the moment of inertia changes from I sub I to I sub F, the conservation of angular momentum can be used to find the final angular velocity in terms of the initial angular velocity. I sub I, W sub I equals I sub F, W sub F equals constant. And Good morning, it's Dr. Will, CBMD, PhD. This is Wednesday, November 9, 2011, 1:04 a.m. Eastern Time. Quick note. If a uniform body of circular cross-section rolls on a rough surface without slipping, the velocity and acceleration of the center of mass are simply related to the angular velocity and angular acceleration according V sub c equals RW, A sub c equals R alpha. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 1:05 a.m. Eastern Time. Quick note. The total kinetic energy of a rigid body rolling on a rough surface can be expressed as the sum of the rotational kinetic energy about the center of mass and the translational kinetic energy of the center of mass. K equals 1/2 I sub c W to the power of 2 plus 1/2 mv sub c to the power of 2 n. Good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 1:06 a.m. Eastern Time. Quick note. If a body rolls down an incline without slipping, one can use conservation of energy to find the velocity of the center of mass as the body falls through a vertical distance h starting from rest. The result is given. From this expression, one can also find the acceleration of the center of mass. V sub c equals, in bracket, 2gh divided by 1 plus 1 over c divided by mr2, in bracket, closed, 1/2 n. Good morning, it's Dr. Will, CBMD, PhD. This Wednesday, November 9, 2011, 2:49 a.m. Eastern Time. Quick note. If you do not understand interferometry, it is basically the same system used in some laser systems to produce holograms and spectrum analysts. I'm only dealing with generating harmonics and subharmonics for research only. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 2:50 a.m. Eastern Time. Quick note. Rice's microscope may have worked on the principles of using interferometry, and this could be why. To this day, the microscope is not understood. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 3:01 a.m. Eastern Time. Quick note. Rice's first machine used two transmitters. These two transmitters produce a mixing of frequencies, F1-F2, and the addition of some frequencies, F1 plus F2. In the mixing process, many waves are produced with many fundamentals. The scope pictures above show the mixing process produces. And good morning, it's Dr. Will, CBMD, PhD. It is Wednesday, November 9, 2011, 3:03 a.m. Eastern Time. Quick note. These wideband radio waves must contain all the fundamental frequencies for the known virus. The transmitters are then adjusted to produce the correct fundamental frequency. This is done by adjusting the amplitude output of one of the fundamental oscillators, which change the fundamental frequency and creates many harmonics of other fundamental frequencies, one behind the other, slightly out of phase. An example of this would be to take the 62 Hz at 3 volt peak to peak and 1.059 kilohertz at 5V peak to peak, and mix the two to get a total output of 1.5 volt peak to peak, getting the oscillator at 150 MV, and then modulating the 1.6000 MHz transmitter. The frequency ends up at 1.55620, the fundamentals with all the harmonics added. Can you see? And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 3:06 a.m. Eastern Time. Quick note. More than 75% of the organisms Rife could see with this universal microscope are only visible with ultraviolet light. But ultraviolet light is outside the range of human vision. It is invisible to us. Rife overcame this limitation by heterodiming, a technique which became popular in the earlier radio broadcasting. He illuminated the microbe, usually a virus bacteria, with two different wavelengths of the same ultraviolet light frequency, which resonated with the spectral signature of the microbe. These two wavelengths produced interference where they merged. The interference was in effect a third longer wave, which fell into the visible portion of the electromagnetic spectrum. This was how Rife made invisible microbes visible without killing them, a feat which today's electron microscope cannot duplicate. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 3:16 a.m. Eastern Time. Quick note. In the expression for torque T equals R times F, is R equal to the moment arm explained? Answer, no. The vector R is the position vector of the applied force relative to some origin. The moment arm is a projection of R along the direction perpendicular to F, whose magnitude is the perpendicular distance to the line of action of F from the axis of rotation. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 3:17 a.m. Eastern Time. Quick note. Can a particle moving in a straight line have non-zero angular momentum? Answer: no. A particle has angular momentum about an origin when moving in a straight line, as long as the line of motion does not pass through the origin. Its angular momentum is zero if the line of motion passes through the origin, in which case r is parallel to v, and L equals r times p equals r times mv equals 0. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 3:18 a.m. Eastern Time. Quick note: if the torque acting on a particle about an arbitrary origin is 0, what can you say about the angular momentum about that origin? Answer: since t equals dl divided by dt equals 0, we conclude that L is constant to the motion. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 3:19 a.m. Eastern Time. Quick note: a particle moves in a straight line, and you are told that the torque acting is 0 about some unspecified origin. Does this necessarily imply that the net force on the particle is 0? Can you conclude that its velocity is constant? Answer: no, the net force is not necessarily 0. If the line of action of the net force passes through the origin, the net torque about the origin is 0, yet F sub net equals 0, therefore you cannot conclude that its velocity is constant. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 3:21 a.m. Eastern Time. Quick note: suppose that the velocity vector of the particle is completely specified. What can you conclude about the direction of its angular momentum vector with respect to the direction of motion? Answer: since L equals r times p, L must be perpendicular to the direction of motion. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 3:23 a.m. Eastern Time. Quick note: if the net torque acting on a rigid body is non-zero about some origin, is there any other origin about which the net torque is 0? Answer: yes, if the origin is one for which the moment arm is 0. The net torque will be 0 about that origin. In fact, any origin for which r times f equals 0, that is r parallel to f, will satisfy this condition. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 3:24 a.m. Eastern Time. Quick note: if a system of particles is in motion, is it possible for the total angular momentum to be 0 about some origin? Yes, the vector sum of the individual angular momentum can be 0. For example, the two identical particles shown at the right moving in circular orbits, but in the opposite sense, have angular momentum which cancel each other. L sub 1 points out of the page while L sub 2 points into the page. Also, L sub 1 equals L sub 2 equals mvr. Decimal. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 3:25 a.m. Eastern Time. Quick note: a ball is thrown in such a way that it does not spin about its own axis. Does this mean that the angular momentum is 0 about an arbitrary origin? No, angular momentum is defined as L equals r times p equals r times mv. As you can see from the diagram, the particle has non-zero angular momentum about an arbitrary origin, even when the particle moves in a straight line. L equals 0 only when r is parallel to v, or when v equals 0. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 3:27 a.m. Eastern Time. Quick note: when a cylinder rolls on a horizontal surface, are there any points on a cylinder that have only a vertical component of velocity at some instant? If so, where are they? No, the diagram shows that every point has a horizontal component equal to r sub w, where r is the radius of the cylinder. However, points along a vertical line through the center of mass have only a horizontal component of velocity. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 4:59 a.m. Eastern Time. Quick note: drinking water from plastic pipes is harmful. Science Daily, November 8, 2011. Pipe and pipe systems are now commonly used to distribute water in many Norwegian homes. The inner pipe for drinking water is made from a plastic called cross-linked polyethylene PEX. Are these pipes harmful to health, and do they affect the taste and odor of drinking water? And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 5:01 a.m. Eastern Time. Quick note: previous international studies have shown that plastic pipes can release substances that give an unwanted taste and odor to drinking water. It has also been suggested that some of these substances may be carcinogenic. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 5:02 a.m. Eastern Time. Quick note: the aim of the study by the Norwegian Institute of Public Health was to investigate whether leakage products from these pipes are harmful to health and if they affect the taste and odor of drinking water. These leakage products consist of residues of additives used during production to give plastic pipes their desired properties, as well as any subsequent breakdown products. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 5:03 a.m. Eastern Time. Quick note: the study showed there are no health risks associated with drinking water from PEX pipes. A few types of PEX pipe may cause prolonged undesirable taste and odor if the water remains in pipes over time. Although the taste and odor usually dissipate with use, Water from 2 of the PEX pipes still have had an unpleasant smell and taste after a year. The level of volatile organic compounds that leaked from the new PEX pipes was generally low. The level was further reduced with use. No correlation was found between production method and leaking products. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 5:04 a.m. Eastern Time. Quick note. 10 different types of PEX pipes available in North Asian market were tested for leaching products in the standardized laboratory test. The water was in contact with the tubes for 72 hours. 3 different manufacturing methods produce pipes known as PEX A and PEX B or PEX C. These methods use slightly different additives. But this study found no correlation between production method and leakage products. And good morning, it's Dr. Will, CBMD, PhD. It's Wednesday, November 9, 2011, 5:05 a.m. Eastern Time. Quick note. 2.4 di-tert-butyl phenol, methyl tert-butyl ether, MTBE, were 2 of the most commonly occurring substances detected in the water in the experiments. For 3 types of new pipe, MTBE was detected in higher concentrations than the U.S. government's recommended limits for taste and odor of drinking water, U.S. EPA, but the values were reduced to below this limit after the tubes had been in use for a while. And